I'm joined by Dr. Anna Sanchez Avila, who is a PhD student at the University of Dundee, and Rebecca Williams, who's also a PhD student in the MRC Cognition and Brain Sciences unit at the University of Cambridge. And there you go. That's who they are, and that's what we're going to be talking about. We're going to talk about MEG scans and super microscopes to understand frontotemporal dementia over the next 30 minutes. Hi, both. Thanks for joining.
Hello.
Hi.
Pleasure to be here.
And I'm
Yeah,
6 minutes. You've got the pressures off because you've now got 6 minutes less. I don't know if that's a good thing or a bad thing. I'm sure both of you could easily fill half an hour all by yourself. So why don't you introduce yourself? Rebecca, why don't you go first?
Yeah, hi. So I'm a second year PhD student, so just edging into my second year now, which is very exciting. I did my undergraduate and my master's at the University of Oxford in psychology. I got my start, and then moving over to neuroscience because I found I liked that biology side of things. And on the weekends, I like making candles and playing Dungeons and Dragons because I'm a massive nerd.
And also a massive traitor, apparently.
Yeah, I know, defected. Or saw the light. You've got to pitch it depending on like who you're talking to.
I'm in Oxford, and there's only one way to look at this. And Anna, why don't you introduce yourself?
Yeah, so I'm Anna. I'm a last year PhD student, due to submit in 5 months. So
I don't know if that makes you all smug when you look back at Rebecca, or Rebecca's looking forward going, oh,
I'm just jealous.
It's just that kind of final home stretch now. And I'm based in sunny Scotland in Dundee. And my background is mostly looking at motor neuron disease, ALS. But as well now kind of the overlap with frontotemporal dementia. And I mostly use very fancy microscopes on a normal day-to-day basis. And that's kind of mostly about me. As my Twitter bio says, if not researching, I'm probably going to be dancing around somewhere. So
and have you been in Dundee? I mean, it was really important to me actually that we have, because they really go, oh well, they're PhD students, so what? But it's really important because actually the majority of research that's done actually is done by PhD students, right? No disrespect to the more senior people that have joined us across the day, but the kind of the day-to-day, the legwork, there are a number of PhD students far outnumber the number of postdocs or professors working in places. So whilst they might provide that overall leadership and that air cover, that you're the ones that kind of do the real work and are discovering along the way, which makes you so interesting and passionate to talk about because you're so enthusiastic about your work as well. Rebecca.
Yes.
Wait a second. You're the one that's using those clever scanners that have a chair and they're like being in the old
it's a 50s hairdryer. Yeah, exactly. That's how I describe it to everyone all the participants that come in.
So tell us all about your work.
Yeah, so like you say, I study frontotemporal dementia, which is one of the rarest types of dementia, and it's unique in a couple ways. One is that it's a young onset dementia, which means whereas in Alzheimer's disease, most of the people that we see are going to be over the age of 65. Most of the people that I meet on a day-to-day between the ages of 50 to 65. So it happens a little bit earlier. And whereas a lot of dementias are associated with memory, I think is the thing that we really link to the Alzheimer's in particular. In our patients, their memory is pretty fine. We instead see behavioral changes. So in particular, they get apathetic and impulsive. And I'm interested in the apathy in particular. And I like to describe apathy as their get up and go, got up and went. It's a problem with motivation, that they're not very goal-directed anymore. And that's the real problem, both for the person with frontotemporal dementia, because we know that apathy is associated with things like faster cognitive decline, loss of independence, and even mortality. You know, people with apathy simply die sooner, which is definitely a problem. So we hope by targeting apathy, we can improve apathy and the overall disease trajectory for these patients. But also it can be really difficult from a loved one caregiver perspective to see someone just kind of sit in a chair all day, not doing anything and seemingly not caring. There's actually a paper which was released about these kind of caregiver experiences that was called "He Just Won't Get Out of the Chair and Do Anything," which I think was a really kind of powerful headline of showing just how frustrating it can be. So there are lots of different ways that we can look at apathy. We in clinics use a lot of questionnaires and what we call self-report measures of asking people how apathetic they are, and they can be really helpful. But some of the research that we've done recently shows that especially in these patient populations, they might not be particularly reliable or meaningful. So we're really interested in that biology side of things. We're interested in what makes people do things, or in our case, not do things. And we don't think it's as simple as problem as they can or can't do it. A lot of the people that we see are still very mobile. They can be relatively fit and healthy in other ways. So they could do these things. They could continue these hobbies and activities. And we don't even think it's necessarily a problem of enjoying. I think if you ask people what's the most basic reason you can think of to not do something, it's because you don't want to. But our patients aren't depressed. And if you ask them, which, you know, they're the most important ones, they're the ones living with it, they won't tell you that they don't enjoy things anymore. I once had someone playing a game with me and they went, oh yeah, I got 1,000 points. But they still got up and walked away because they couldn't be bothered anymore. So it's this weird kind of problem. And traditional theories of apathy mainly look at that kind of reward, this idea that people don't enjoy things anymore, so they're not motivated to engage with the world. But we don't think that theory is particularly great in that, one, if you ask the people with frontotemporal dementia, they'll tell you they still love doing all these things. And two, that dopamine is the chemical in the brain that we mostly associate with reward. And therefore, you'd assume that if you improve dopamine, then you'd improve apathy. And we see that dopaminergic treatments aren't all that effective against apathy. So as part of my PhD in my lab with my supervisor, Professor James Rowe, and past PhD students like Frank Heisman, are looking at developing a slightly new theory of apathy, where instead of looking at reward, we're instead looking at it in the context of the Bayesian brain. You may have come across Bayes theorems in high school or in college. Bayes is all about probabilities. So we're interested in how your brain updates probabilities of things going on around you and how it integrates how you can change those probabilities by acting with the world around you. So my favorite example of this is what I call the birthday analogy. If you wake up on your birthday and you have a very clear expectation of, I need presents, I need cake, I need to blow out candles upon the cake, I need everyone to wish me a happy birthday. And then you run downstairs and in a full 16 candles moment, everyone seems to have no idea that this is your special day. And there's no cake and there's no presents and no one's wishing you a happy birthday. If that happens, there's a big disconnect from what you expected and what is happening. And there are two ways you can fix that. You can either update your belief, you can update your expectations and say, well, you know, Rebecca, you're 23, maybe you're too old now for everyone to stop what they're doing for your special day. Or you can actively try and change the world around you in order to meet those expectations. You can go to everyone and say, excuse me, don't know if you knew this, it's my birthday
I need
today.
you to make a bigger fuss.
Or you could even go to the shop and get your own presents, right? So those are the two ways that you can deal with that. But if we instead turn that on its head and look at someone who is apathetic, and we say they wake up and they don't really have a clear idea of what to expect. They know that maybe they unwrap something, there might be food involved, but it's all a bit wishy-washy, it's all a bit noisy. And then they go downstairs and no one wishes them a happy birthday, there's no presents, there's no cake. They don't need to update their belief because it was noisy enough as it is. There's not a big deal. There's not a big difference between what they expected and what they see. So they don't go get their own presents. They don't remind people it's their birthday. They don't even update anything. They just walk out the door seemingly completely uncaring that everyone's forgotten it's their birthday. And that's how we want to conceptualize apathy. It's this lack of precision, this lack of confidence in how you expect the world to be, so that the world can change in any number of ways, but it's still close enough to how you expect it to be, which means there's no motivation to do anything to change it. And we're particularly interested biologically in a chemical called GABA in the brain and how this might link in with our theory, which is where the MEG scanner comes in and where our modeling comes in. So I like to think of the brain as being a set of radios, a set of walkie-talkies. And when the walkie-talkies are on the same frequency, they can communicate really clearly with each other, no problem. But if they're on different frequencies, the communication gets really noisy, gets really crackly, if they can communicate a message at all. So if you imagine a brain in which these radios are on different frequencies, then there's no way you can communicate a really precise, confident expectation of the world when the apparatus that you're using is so noisy. And we think GABA, this chemical, is important for tuning the radio. And we know that in people with frontotemporal dementia, GABA drops. They have less GABA. So we're using brain scanning, both models, Bayesian models and physiological models of the brain, and doing behavioral tasks, all combined together to try and understand, is this a valid way to think about apathy? And by targeting GABA, could we produce new, more effective treatments for apathy that would improve that disease trajectory and let our patients live better and live longer?
Wow. Cool, Anna, you've got to follow that.
I know. Big challenge.
That was amazing. That was probably the best description of apathy I've come across. And
School.
you so brilliantly kind of gave examples of how people experience that. And is apathy something that only people with FTD experience, or is this present in other forms of dementia as well?
Excellent question. So we think that apathy is not only present in other, well, not we think, we know, apathy isn't only present in other forms of dementia, but across neurology and psychiatry more broadly. So depression, we often get apathy and depression as well. So apathy is a really common problem, not just in FTD, not even just in dementia, but really broadly. And so it would be really cool to find a unifying theory of apathy. But at the minute, we're just focusing on our frontotemporal dementia patients, and past research, which looked at this kind of new theory, has looked at it in Parkinson's disease as well.
Which potentially has some real benefits, doesn't it? Because if apathy is more highly present in people with frontotemporal dementia, then I guess it gives you more of a way to zero in on the right population of people. And then you can test out, if you find the cure to apathy, you can test it out in other people as well to see if it works there, or was this unique. But I guess could that be because there are different reasons for apathy, or are we just purely talking about the chemicals and the things going on in the brain? Are they the same, whether your apathy comes from depression or whether it comes from FTD? Is it the same underlying biology?
Yeah, so another really great question. And I guess the short answer is we don't really know. So transdiagnostic research, which is research where we're not just looking at a particular patient group, but saying, no, the symptom is present everywhere. We need to collapse across all these boundaries that we've been setting up to look at it across and see if we can find a solution that goes beyond the boundaries of this particular patient group, is relatively new. So I think it's a really exciting idea that we could collapse across all these boundaries and find one theory, one chemical in the brain, one circuit in the brain, one treatment that can target apathy across dementias and across depression and everything else.
Because I guess that'll also help people who, stop eating or doing other things that we know that will help them.
Yeah, that would be great, but we're not sure.
I'm going to let Anna give us a run through now. Don't worry, Anna. Just tell us
it's
about.
Okay, very big shoes to fill, but I'll try my bestest.
Tell us about your work.
I'm kind of looking on the flip side of that. So I mostly, as I said, work on motor neuron disease. But what's very interesting is that we now know that motor neuron disease, which is kind of a most motor symptoms. So if you've probably heard the news, sadly, W. Weird passed away very recently due to MND. So we know that MND kind of exists here with symptoms that make patients unable to move, and that relates to walking, talking, and sadly, eventually breathing. And then we have frontotemporal dementia on this other side of the spectrum, which Rebecca just brilliantly explained. And then we have people that exist kind of somewhere in the middle. So either they have very clear frontotemporal dementia symptoms with some motor symptoms scattered throughout, or very clear MND symptoms or ALS symptoms with some sort of cognitive changes that we didn't know before were common in ALS. So mostly what I focus on is to study brains of people that exist kind of in that middle spectrum and people that exist totally in this side of the spectrum, to see if different parts of their brain are different. And what I'm looking at is connections, so brain connections. Our brain cells communicate with each other in what we call synapses. So basically you have one cell talking, the other one listening, and then it acts on what it's heard before. So things like movement, there's one cell that says, okay, we need to move to pick up something. Then the other one goes, okay, if I need to do that, then I need to tell my arm to extend, to reach, and then contract to get back. So you need that communication to work in order for you to move. If that communication stops, then there's no movement. But in theory, your ability to know what to do is still intact. However, the people that are in the middle have more symptoms than just that. And we want to know what the difference is between those two. And one of the reasons why is because, unfortunately, people that exist somewhere in the middle have an even worse prognosis than people that are just on this side. And also, as a kind of patient and caregiver, it's also important for you to know what to expect when it comes to planning and processing. So that's kind of the big picture of why we're looking at it. The problem is the connections that we look at are tiny. So we have more synapses, those connections in our brain, than there are stars in our galaxy. So we're talking billions and billions, like so many zeros. So that makes the job quite tricky. And also they're insanely small. They have to be for them to fit all in one little head of ours. So that proves a challenge when it comes to looking at them. So we need very advanced microscopy skills for us to be able to look at them. In my case, I mostly just look different areas of the brain, and right now at this stage I'm just counting connections and seeing, okay, does this area of the brain have more connections, less connections? Is it different between the people that are in the middle of the spectrum onto one side? So to do that, we use a technique called array tomography, and that is kind of the microscopy technique that we use, and it relies on brains from donors that have donated their brains to science. And we've processed it, and we need to cut it very thin. So if you imagine your human hair, normal human hair, you thin it out 100 times, not lengthways, sideways. And that is the thinness of the tissue that we need to cut. So I'm working with basically see-through material, then staining it in fluorescent colors and looking at it down the microscope, and it makes it light up and look like a galaxy. And I take those images and put them into my trusty computer that counts them for me. So that's very handy. And that's kind of mostly what I do in my day-to-day life.
Wow. I mean, I love that there is this kind of connection between your work. I mean, that's just incredible. You must need a steady hand, right?
Yes, a steady hand and a lot of patience is definitely.
My eyesight just wouldn't work for that. I mean, this is fascinating. I mean, this is proper sci-fi stuff between both of your work as well. I mean, whilst they're very different, you can see that there is this underlying connection, because at the very end of this, what you're talking about is somebody's willingness or engagement to engage, to stand up or extend their arm, because they're just not going to. And yours is that their brain tells them to extend their arm. It just doesn't do what you want it to do. So it's interesting. There might be connections there down the line. You talked before about the people in the middle. So is this people with very, very early motor neuron disease? How does everybody? So there's a question that I'm sure people would ask. Does everybody who gets that, because getting a diagnosis of motor neuron disease, is notoriously quite tricky, isn't it? That it's not the first go-to thing that people are told. Does everybody get motor neuron disease differently, rather like they do with Alzheimer's disease? We're not really going to talk about MND a lot today, but there's real connections, and the Dementia Research Institute looks at MND as well, or ALS if you're in the US.
Yeah. So there's clear connections, especially with frontotemporal dementia, to the point where now we see them as a disease spectrum. So basically, we're talking 50% of patients that get diagnosed with MND will end up developing some level of cognitive problems, and around 15% will reach a full-blown FTD diagnosis, as well as the ALS diagnosis.
15% of people with MND.
And the other way around. So around 50% of FTD patients, besides the symptoms that Rebecca mentioned, they develop some level of motor problems with movement, and then 15% end up reaching a full ALS diagnosis. So there's a clear overlap, and it's crucial that we understand it, because then we can understand the actual mechanisms. If we understand what they have in common and what they don't, that could give us huge insight into the actual mechanisms behind the disease.
Is apathy a symptom that people with motor neuron disease experience early on in their disease?
So I wouldn't know about early on, but a group in Edinburgh led by Sharon Abrahams actually developed what's called the ECAST score, which is a cognitive and behavioral impairment test that's been designed to kind of correct for the problems that ALS patients might have. So basically, when you can't move or talk quite freely, you need to do some adaptations when it comes to testing and to kind of account for that. And they've done that, and they've seen that in those 50% of patients that have some level of cognitive and behavioral impairment, apathy is actually quite high up there. So it shows the clear overlap between diseases in this stage.
I love both your work. It's got so much. I mean, I've got questions here about what's the potential impact and what's MEG. But honestly, I think the what I love about both your work is that it's kind of nicely on that, in that space of it's very much hard science, but has some real practical implications as well. That this isn't something that we're just, I think all too often some researchers are kind of accused of kind of doing research that's just of interest to you. I think, and we're constantly reminded that we should only, the research should have some real world impact, that people living with dementia or motor neuron disease, this should be something that they also think is important. And I can't help but think that anybody who's living with dementia wouldn't say that what you're doing has some real potential benefit there. But I mean, if you can cure apathy? I don't know. How far away? So this is, I know we're coming up to our time as ever. I always run out of time because I've got a question to ask. Actually, I'm going to just do this question. You're both PhD students. Has anything surprised you about your work so far? And you both spoke so passionately and excited about it. It's clear that, you're still excited about your work. But what surprised you, Rebecca?
So I think I've always really liked the idea of doing, let's say, clinical work that has real world applications. So when I came in and started working with people who have frontotemporal dementia and their families, and even a lot of the healthy controls that we have who are in because they know someone who has dementia, I knew I'd enjoy that, and I did. It's amazing to work with them and see firsthand the kind of impact that research has, and get the stories from their lives of what they're living with. I think one of the things that surprised me is how much I've enjoyed the technical elements of my PhD. So I'm a computational
nerd thing you said at the start.
Back to the nerd thing at the start. So I'm a computational modeler. So I build models of the brain, dynamic causal models, to help simulate and understand where all of these wiggly signals from the MEG are coming from and what could be causing them. And it's a lot of coding. It's a lot of reading algorithms and mathematics. And I didn't expect to enjoy that as much as I have. That's been really fun. And I think the most heartwarming surprise I've had is coming from undergrad psychology. The reason I participated in research studies was because of the money or the course requirements. And to go from that,
there's no money.
There's very little money. £10 an hour is a lot to an undergrad. But going from that to our participants, who 9 times out of 10 will say that they're there to help because they know that this research needs to be done, because they know it probably won't help them, but it will help someone. And I think that's just so admirable. And it's the same, like, say, for the people with frontotemporal dementia, for their loved ones and their caregivers, but also for the healthy controls who have relatives a lot of the time with dementia. They're here because they want to help. And I think that is just really heartwarming, and I think they'll never know just how much it means to us that they come in and do the research and give us such wonderful high quality data.
And because it's some of the rarer forms of dementia as well, there are definitely not enough people, particularly with Lewy bodies and FTD, coming forward to participate in research. So if these diseases are affecting you or your family, please do go sign up to join Dementia Research, and it helps Rebecca.
It is greatly appreciated so much.
To find the people that they need for their research. Anna, what's exciting you? I mean, apart from, are you excited to try and get those samples even thinner than they already are?
I don't think that's even
Would that
possible. But I think that what surprised me the most is how much I'd enjoy the actual day-to-day, like little experiments rather than the kind of big picture, and just like seeing the beautiful like light up images of the brain, and like how you can change colors and just fully make it look like a galaxy, like outer space. Just kind of finding joy in those little moments.
Who knows that isn't? I mean, this is all a simulation anyway, right? I mean, this is I'm going to let you both go because my next batch of guests. Rebecca, go have a look at the new chip project, NEU chip. It's about how they're using stem cells integrated with microchips to grow 3D brain models onto microchips so they can plug them into computers. It sounds like it might be something that's
I'll definitely have a look at that. Thank you.
Thank you very much to my brilliant guests, Anna Sanchez Avila and Rebecca Williams from Cambridge University.
Thank you so much.
Thank you.
And Rebecca also as well, you might, and Anna, you might want to stick around because Bridget is quite influential when it comes to fellowship funding.
You're very kind.
both of your research studies sound exactly the kind of things that Race Against Dementia and the other charities as well, of course, that gets them excited. So when you're looking for your fellowships, do keep these guys in mind. Thank you very much.
Bye.
Have a great rest of the day.
Bye.
Okay. Now I've been playing videos at every half an hour, and I can't now because who would I choose? Which video would I play? I can't play one film over the other, and so that would be unfair of me. Instead, what I am going to do is to remind everybody who's watching that the whole point of today is not only to share the research we've been talking about with our incredible researcher guests, but also to raise money for four charities who we're going to be talking to over the next half an hour. Race Against Dementia, the Lewy Body Society, Alzheimer's Society, and Alzheimer's Research UK all fund research and do a million other things as well to influence policy, to help people living with dementia and to eventually beat this disease. Please do give what you can. Every penny helps, and it's at chatathon.uk/donate, and the money we raise today will be equally divided amongst the four charities.